Hello, everyone. Warmly welcome to the NMR series is organized by the Treesearch Research Infrastructure. My name is Anastasia Ryasanova, and I'm coordinator of the infrastructure. And it's my pleasure to welcome you on my behalf and on the behalf of Professor Gunnar Westman, who is co-organizing the NMR webinar series together with me. And today it's a great pleasure for us to welcome Professor Heiko Lange, who will give you the second webinar on the NMR field. So please, Professor, the floor is yours.
Yes, thank you very much, Anastasia. Thank you very much, Gunnar, for the invitation. And I'm very happy to be part of this webinar series on the NMR analysis of the, in my case, of isolated lignins. The techniques that I will show you, the HSQC and the phosphorus NMR are generally, and that's an important point, are generally applicable to polyphenols. So I'm talking about lignins, but whatever I tell you, you can eventually adopt also to the field of tannins, or even eventually to lignans. So please keep it in mind that I'm talking about lignins, but it's generally applicable. First of all, obviously, let's record a little bit what are lignins. I mean, because it's a very special type of natural product, and it will pose a lot of issues, as you will see, and that is obviously important to know right from the beginning, because otherwise I design my analytical approach in a completely wrong way eventually. So the lignins that I'm talking about are coming mainly from waste biomasses like straw, or from trees directly, in form of lignins that come from paper industries, where the lignin is the byproduct from the cellulose production. What we normally get in the laboratories as isolated lignins are these typical brown powders, depending on the isolation process, more brown, more yellowish, etc. But they are definitely colored, and the color comes, and that's another important thing, doesn't come from the perfect untouched lignin structure, because that's not a chromophore, right? There are no chromophoric groups in a lignin that would create a color. But the color indicates a degradation of the lignin structure, impurities in general. So what we want to do ideally in the following is to understand the lignin structure and also the impurities, as you will see. Then all these lignins come sooner or later from a biorefinery. Just to give you a background, right? As I said, we take the waste biomasses, and then they are treated, the waste biomasses or the wood, directly in the pulp and paper industries. So we can see everything as some type of biorefinery, where one of our products is then the lignin. When we get the different biorefineries and have a closer look at them, we can see that they are differently organized, obviously, right? Depending on the need that we might have pre-treatments, we might have the classic pulp and paper production processes, and we might have novel processes that aim for the biofuel production, etc. The interesting thing in all this is that we have our lignin as a product that has in its structure a recording of the process that has been used for isolation. And this is one of the major problems if you think of a valorization of the lignin. And it is one of the major issues that, apart from the natural differences in the lignin that come from the biomass source, we have the additional differences that come from the processing. So talking about a lignin structure and then placing always this image from the 60s is probably not a good idea, because this image that we have here is eventually a correct image for a very particular type of lignin, but it cannot be seen as a general image of lignin for all the lignins that come from all the different processes of biorefinery, pulp and paper, etc., etc. So before we can even think of a valorization of the lignin as one of the parts of the biomass that has been fractionated and taken away from the other parts of the biomasses and isolated in more or less pure form, we need to get a better picture of its structure. So we need to make really an analytical effort to understand whether this is a correct image or whether this image is eventually completely off in terms of molecular size, in terms of functional units that we have, etc., etc. So we need to think of the structure to describe what we have as a brown powder. Because without the structure, we cannot devise a certain lignin for a certain valorization. And this is the key point at the end of the day in order to get the green transition done, that is especially in Europe now postulated or demanded by the governments, etc., etc. So it is not set from the very beginning. It is not clear whether every lignin is suitable for every type of valorization. And if we want to make use of the biomass that is a renewable resource, but that as such is also due to some limitations because it needs time to grow, we have not endless land to plant it, etc., etc. So we must use it in a very, very intelligent way at the end of the day. So we should not waste it by using the wrong type of biomass, the wrong type of fraction of biomass for the wrong type of valorization. So in order to get that done in the best way, we need to understand the structure. And I always tell my students that, and it works rather well, they should ask themselves 4 questions regarding the lignin structure. So first question, what can I know? What can I know about the lignin structure? What can I know, actually? What can I know with my equipment, my laboratory, etc.? And what can I really know? So what is the real knowledge that I can take out? What is maybe a suggestion by the analytics that I do, etc.? If I answer these 4 questions in an honest way, I can eventually arrive at a point where I can make a prediction in terms of this is my lignin, this is the structure, and then on the basis of that, I can say this is a potential valorization effort for my lignin. How do we get that? And what does it lead to? Well, if you type lignin as a lignin structure into Google, you get all these types of images because everyone working with lignin does his or her type of analysis and comes up with his or her interpretation of the structural features. There are some things that are not discussed anymore. There are lots of other things that are still under discussion because our analytical methods are, let's say, not perfect in a certain sense, but we have to make an effort in order to combine different types of analytics. So the different types of analytics that we generally need to combine and that go hand in hand together with the 2 that I will be talking about mainly is seen on this slide. So I'm talking about the HSQC, I'm talking about the phosphorus NMR, but you need to see it together with molecular size analysis, eventually with thermal data, because that might give you a certain hint how to interpret some data that you have. And we need something like eventually elemental analysis, log PE studies, if you like, and if you need that information for your application also, and XPS, for example. So all types of analytics that give us a hint of the structure, and that one helps normally the other in a way that we can interpret better the data that we see in order, because as I said, the analysis methods are not perfect, and we sometimes have can have 2 good explanations for a single feature that we see. Now, as I said, today I focus on the quantitative HSQC or a quantified HSQC, and we will see what is the difference, and on the quantitative phosphorus NMR, and on the features we can understand from that. Now, let's start with the simpler one, the phosphorus NMR. So it's a heteronuclear NMR measurement. Obviously, it's not the standard proton or standard carbon NMR. And the first question that is obviously for those of you that are new to the field, for some of you might very well know. So the next 2 minutes maybe are less interesting. For those that are new, it is the first question that comes obviously to one's mind is, why do I do a phosphorus NMR if I'm dealing with a natural structure that doesn't contain phosphorus? First thing to do, therefore, obviously, is a functionalization of my lignin or of the polyphenol that I isolated. The functionalization is normally done in this field by this reagent here. This is a tetramethyl phospholan, and this molecule here, activated by the chloride that sits on the phosphorus, gives us practically a phosphorylation of all the OH groups, independent of the character of the OH groups. So we can phosphorylate primary, secondary, and even the phenolic OH groups. Running the correct protocol, we can be sure that this phosphorylation is more or less is quantitative on all the different types of OH groups. Once I have phosphorylated all the OH groups, I end up with a structure that has I didn't touch the carbon structure of my lignin. I just I just manipulated the OH groups, and I create for each of the different types of OH groups, so primary, secondary, aliphatics, and phenolics, and also the acidic OH groups on the carboxylic acids that we might have in the lignin, I create different environments for the phosphorus atom. And I can do, therefore, nicely a phosphorus NMR, because then in this phosphorus NMR, I only see the phosphorus, and I do not have all this structural variety that comes in from the direct surrounding of the OH groups, right? If I would do a proton, I get a forest, very, very low resolved, and I cannot interpret a lot. If I do the phosphorylation and I run a phosphorus NMR, I only see the heteronucleus that I introduced in very specific positions. So I'm basically hiding a huge part of the problem of the structural diversity in a first-hand type of analysis, so that I get an entry into my analysis protocols. So after this phosphorylation with this molecule, with this reagent here, I get a phosphorylated lignin that I can then treat with a standard phosphorus NMR protocol. The phosphorus NMR protocol has been debated, but at the end of the day, if you run it correctly, you get reliable data. There are lots of pitfalls where you can make mistakes, but running it correctly gives you really a nice tool for getting along with a structural identification. On this slide, without going into details, I just re-report from this publication the key aspects, and those shouldn't be changed, because if I change my parameters of acquisition in the NMR, I change eventually my NMR outcome, and that is one of the points that will come up once in a while during this seminar, that you have to have a certain type of standardization. If you don't have a certain type of standardization, and if you do not make sure that the quality of your spectra meet a certain level, then obviously we enter in a type of discussion where we do not talk on the same level. So that makes things very, very difficult. So therefore, I highly recommend that we stick all to one protocol. Now, it's maybe not my preferred protocol, but it's a protocol that everyone uses, so I keep comparability basically across the works, and that is, for making progress, very, very important, obviously. So the two key points here is to use an inverse gated decoupling pulse. That means I cannot use the standard pulse that Bruker has as a very, very standard. I need to do this inverse gated in order to make sure that I do not over or underestimate some of the responses during the NMR acquisition of the phosphorus, because obviously when I do the standard NMR technique, this phosphorus here feels the other nuclei obviously in its surrounding, and they feed back eventually magnetization effects, and we have to get rid of those artificial differences. Or we have to eliminate these differences in a way that we do not have artificial over or underestimation of the abundance later in the spectrum. Then relaxation delay. You have to be patient running a phosphorus NMR. It's not as quick as a proton NMR, obviously, right? But if you do the right relaxation time, you get good spectra. Run a certain number of scans, because otherwise we have a very unsatisfying signal to noise ratio. If we run the phosphorus NMR, it should be run in a way that the spectral window that we have in the NMR shows us 3 control peaks, basically. First control peak is this one, on the very right of this spectral window that I showed here. This is excess reagent. So that means I must see during the analysis the excess of this reagent here, because that ensures that I had put in enough in order to really phosphorylate all my OH groups. If I don't see the excess, I cannot be sure that the phosphorylation has been quantitative, with all the consequences for my analytical approach. On the very right here, I should see another huge peak, and this huge peak is the peak that comes from the reaction of my reagent with water. So it's another control peak, because I know my lignin is dried, but I didn't dry out the last drop of water. I have some water molecules inside my structure, right? It's not wet, my lignin, but it has been dried to constant weight. The water that stays in at this constant weight is structural water. That is water bound with hydrogen bonding between hydroxyl groups in the lignin, etc., etc. So I must see this. There is no way that I don't see water in a lignin. Okay, so I must see this. And then this little things that I left in the middle is basically the spectra of my lignin. These bumps that you see here, it's not a typical nice sharp signals. These bumps here reflect the structural virality around certain types of hydroxyl groups. This here are all the different types of aliphatics. Okay, they are so different in their nature and their surroundings that I don't get one sharp peak, but I get a bump. And it's perfectly fine for the interpretation, because at the moment I'm happy that I know these are aliphatics. I can quantify the amount of aliphatics. These here represent the different types of phenolics. They are very nicely resolved, and I can say that this region here are the condensed phenolics, or S-type, or condensed. These are the G-type phenolics, and Gs are the H-type phenolics. So for those of you that are not familiar with lignin, lignin is a polyphenolic. It's a phenolic polymer, so a polyphenol, right? So this polyphenol can have different types of monomers. I can have one monomer where I have both R groups being a hydrogen. I can have one monomer where one of the R groups is a hydrogen and one is a methoxy group. Or I can have a monomer where both R's are methoxy groups. Those are the typical natural variants that indicate that vary according to different plant, across different plant types, etc. So I have this natural variation anyway, and it is reflected in the NMR by having different regions for the responses, obviously. Here are the carboxylic acids. This one here is the only sharp peak that I should see, apart from a sharper peak in the acids if I have extractives. This sharp peak here is my internal standard, because I need to quantify against an internal standard. As an internal standard, I have normally cholesterol, or this one here, the norbornene. Both are stable. My internal standard needs to be stable, obviously. The nice thing about the cholesterol, I prefer to use cholesterol for doing lignin NMRs, lignin phosphorus, and I prefer to use this one for doing the tannins, because this one is nicely separated, but still inside my peaks. In case of the lignin, if I do tannins, this one ends up between the OH groups of the tannins. So for the tannins, cholesterol is not okay, and I need to use this one. This is what I just said. Here you see again what it will look like. Imagine this is a lignin, and this is the end of a lignin chain, and we end up with having a beta 4 unit phosphorylated in 3 positions. In this case, this is a Norway spruce, so it should be a softwood, and I can see that it is a softwood. Softwood is typical, only G units, practically only G units, and I see only G units. This is the region for the G. I don't see anything here, I don't see anything there. This is a nice clean spectra of a Norway spruce. This as a background for the phosphorus. Since time is flying, I quickly give you a background for the HSQC. So HSQC is different. HSQC is not a one-dimensional NMR. HSQC is a b-dimensional NMR. And the problem with the b-dimensional NMR is that it is not intrinsically quantitative. Okay, if you see in publications these nice HSQC spectra, no colored, non-colored, okay, those HSQC spectra look always the same by the naked eye. From the appearance of the HSQC spectra, I cannot say whether they have been acquired using a quantitative method or a non-quantitative method. And unfortunately, a lot of people acquire HSQCs using a non-quantitative method and then sell the data as quantitative, because sometimes the fact that an HSQC is not quantitative gets lost, because it's not so obvious why it wouldn't be quantitative. It's not quantitative for the type of measurement that I'm actually doing. It cannot be quantitative if I do not use certain precautions in the pulse sequences, or if I use a detour in order to quantify it in a secondary moment. This detour is this combination of a standard HSQC and a quantitative carbon NMR measurement. Okay, the quantitative carbon NMR measurement is easier to do with the standard set of pulse sequences that come with the NMR instruments than a quantitative HSQC. Here we will go into detail. The other one later. The other one is a so-called quick quantitative HSQC that we used in the past and that we currently still use. It is nice. It is a single measurement, so therefore it's relatively easy. The problem here is that you have to program the pulse sequence yourself, or you have to know someone that is able to do it. The third version is called HSQ0. both of these, the quick quantitative and the HSQC 0, come from a different field. They come obviously from different fields. They come from a field where people analyzed metabolites. Okay, so it has been adopted for the lignin field because at the end of the day, we can adopt it to the polymeric nature of molecules, which is, in case of the lignin, obviously suitable. So it's not intrinsically quantitative, but we can make it intrinsically quantitative running the quick quantitative. If we run a normal one, we correlate it with a carbon NMR. So we run a normal HSQC and we run a nice quantitative carbon spectra. And then you play with Excel after you identified 2 standards in your carbon NMR that you correlate to the intensities of the HSQC. Normally you use one type of motif that you quantify in the carbon NMR, and you use this type of motif for quantifying the aliphatic region in the HSQC. And you use a different type of signal for quantifying the aromatic region. So what is the aliphatic and what is the aromatic region? So here in the upper right, we have the aliphatics. We have the aliphatics. Here we have the so-called anomeric region. And normally, if you see people plotting spectra, as I did here, you see that people plot the aliphatic region together with the up to a 5.5 here, where you enter the anomeric region and you put the aromatic region. Depending on what you want to describe, you can also take all this region here and you enter, you have a bigger picture. You always separate, however, the aliphatics from the aromatics, because the aromatics give you a completely different feedback in the HSQC than the aliphatics. It's not possible to quantify this nice sharp peak here and say this is, for example, 1 proton, and then use a peak here that has the same intensity and claim that it is also 1 proton. That would be absolutely wrong. That is not what the HSQC can do. It's not a quantitative carbon. It's not a quantitative proton. It is a bidimensional measurement, and it requires different types of quantification. So we therefore treat this region alone and this region alone, look for a standard in the carbon NMR that gives me a reference for this region and a reference for this type of, for this region here with these type of structures that come up in this type of, in this region here. So if I do that, at the end of the day, it all breaks down to playing with Excel, and I will not go into detail because obviously you correlate, you normalize the first region according to the aliphatic signal that you chose as a standard, internal standard, and the aromatic region according to the other signal. At the end of the day, as I said, quicker is the QQ-HSQC. If you do not have someone that knows the QQ-HSQC, you run the HSQC 0. Here we use a trick basically, and we run a sequence of 3 HSQCs where we change 1 acquisition parameter. It's a little bit difficult to imagine, but I mean, you run 1 HSQC with a normal HSQC basically, okay? Then you run it again with an incremental change, and you see that the intensity of your cross peaks drop. Because of this incremental change, the response is different. So the signal intensity drops, and you make another incremental change and it drops another time. In any case, you have 3 HSQC spectra with 3 different intensities. Then you start plotting these different intensities and you do linear regressions, and you back extrapolate to an artificial HSQC 0, assuming that the HSQC 0 would have the maximum perfect abundances of the signals, okay? If you do that, you base then your quantitative analysis on this hypothetical HSQC 0 that you get via back extrapolation. In reality, it's running 3 spectra and then playing again with Excel, okay? It's a little bit boring process, but you get a quantitative data from an HSQC. The nicest one so far should be this one, the QQ-HSQC, quick quantitative. This has been, as I said, developed for metabolites, but it is applicable to lignin if you program it well, and if you make sure that you think of the fact that you are dealing with a polymer, so you have to adjust your relaxation times accordingly. Doing this, we basically avoid the the fact that it is non-quantitative, which is on a physical basis seen in this curve here, you see that depending on the coupling constant between the carbon and the adjacent or the attached hydrogen, so probably the things that you want to see, no? I run a 1H13 carbon HSQC, so I see only signals where a proton is connected to a carbon. All the other groups I don't see even, right? But that's a nice thing, because I basically get rid of all the ketones, you know, all the carbons that I'm not really interested in, because they are they contain less interesting information in a first hit, let's say. So I only see HC, H2C, or H3C groups, depending on the coupling constants, which in turn depend on the structural environment of the specific C that I'm looking at, the specific carbon that I'm looking at with its protons attached. I don't get the same transfer efficiency, and since I don't get the same transfer efficiency, I don't get the same feedback. So the quantification cannot be exact, because the different environments cause different effects on the intensity of the signal. So I cannot quantify, as I said, for example, an aromatic CH, which is completely different from an aliphatic CH, because I will have completely different coupling constants. So in the QK-HSQC, I eliminate that via a nicely thought pulse sequence, and I more or less flatten out this region. You see, it's not perfect, right? It's not a straight line, but it's at least much more homogeneous than this bell shape here that you see. Now, applying this method, I can get my spectra. This is an old picture, but it's still a nice picture to this is one of the first ones I analyzed. And the nice, it's always a nice spruce lignin, because we thought it's easy to start with a spruce lignin. Then the nice thing here is that I do not need to standardize via an external standard. I can use intrinsic standards. And the nicest thing about the spruce as a softwood is that I only have this type of, mainly this type of aromatic unit. And I have this nicely isolated proton here. It's a CH. My pulse sequence is made in a way that I can say this is a single H sitting at a single carbon. So I have one intensity 1 for one signal in this region. I can assume that this is directly to be correlated, so I can directly come up with a quantification of the intensities of my cross peaks that I see in the HSQC. So this is therefore the nicest method, because it's the most direct method. I just enter it in a table and I'm done, right? Like I would do for a proton, or like I do for the phosphorus. Doing that and playing with the data that I get from the phosphorus and from the data that I get from the HSQC, I can then make my calculations, putting into correlation the amount of aliphatics and the amount of phenolics and the type of interunit bondings that I can delineate from the structure from the HSQC, and I come up with a picture, a suggestion for the structural features, for example, of a softwood mildwood, okay, or a mildwood softwood lignin. Mild softwood lignin, sorry. Okay, so I get an idea of what the structure looks like, and you can see that this, which is the structure that makes most sense on the combination of all the data in together with a GPC measurement for the molecular size. And you see, you know, if you remember the picture that I showed you, the classical lignin picture from the 60s, right? This is something completely different. We don't have branch structures here. We don't have a huge polymer. This is oligomeric. This wouldn't even be called polymeric, right? So you can imagine that from a chemical point of view, thinking of a valorization in terms of a copolymer, in terms of a material, in terms of pyrolysis, etc., this is something different than the one that I showed you before as a generic, so-called generic lignin structure. So we're talking about completely different worlds, and this is obviously, therefore, thanks to the also thanks to the NMR analysis, a much better entry into a valorization. Now, this is the drawbacks I already told you while talking. So let's say this is our approach. This is how we want to do it, right? We understood in a very quick way. I admit, in a very quick way, how the techniques work and what I can take out. So phosphorus for taking out information on the types of phenolics, on the type of OH groups and their abundance, because it's quantitative. So I know how much aliphatic OH, how much phenolic OH, and which types of phenolics, and how much of each type, and how many acids, for example. And I use the HSQC for taking out information regarding the types of aromatics, in a more, not only the phenolic groups on the aromatics, but on the types of aromatics. Do I have only H? Do I have only S? Do I have a mixture of H and G and S, etc., etc. So, and I get information about the aliphatic region in the lignin because it's a phenyl propanol structure, right? That is something that we get even from the old picture. So I get information by the HSQC on all these nice linkages here, how one aromatic unit is linked to the next aromatic unit. What I don't get is the order, a sequence of linkages. So I cannot say that one type of linkage is directly connected to another certain type of linkage, but I get an overall picture of which type of linkages are present in my lignin, as you will see in the next slides. So I take this approach in order to analyze all these types of lignin that I get from the different types of biomasses across different types of biorefinery processes. If I do that, let's take one step back. If I do that, I would end up with this brown powder here, and I can analyze it. I can do just what I've said, and I come up with a much better picture, like, for example, this one here. If I want to make my life a little bit easier, and especially if I want to see eventually impurities that are in a standard spectra hidden as very small peaks, or as peaks that zooming in, zooming out, I would identify maybe incorrectly as noise, etc. I can do one step in between taking the powder as it comes and doing my NMR analysis. And this step in between is a fractionation of my material. Fractionation of my lignin means I go for different types of solubilities of the different polymer in the sense of the polymer size. I can choose systems that select different solubilities of different lignin molecules according to the number of OH groups that are in there, the number of polar groups, the number of acids, etc., etc. So I can play with my chemical knowledge and try to devise a separation scheme, a fractionation scheme, so that I separate different types of oligomeric structures that all together give me the lignin. Because if you see here, for example, in this, you see already that I have really small structures. And I have that I have eventually some larger structures, right? I have some structures with more phenolics and I have structures with less phenolics. So all this via the various physicochemical interactions that we have in a system where I've tried to dissolve my lignin in a solvent gives me eventually a handle to do a separation of different fractions of all these thousands of possibilities of molecules. Once I have the fractions, I can analyze each of the fractions separately with my set of analysis tools in terms of HSQC or phosphorus NMR. Doing that, as I said, I have the great advantage that I might see in a fraction structural motifs that in the together of everything non-fractionated are so little in abundance that I would not see them, or that I would disregard them as something not worthwhile analyzing because it would be a minority in terms of structural ingredient, for example. And this, now you might say that if it's such a little aspect of the structure that I don't see it if I do the bulk material analysis, it's probably not worthwhile thinking about it. For some applications, that is completely true. For some other applications, that might be the factor that makes the application fail. Because if I have a small impurity with a very, very, let's say, particular chemical characteristic, that might screw up my intended application. So it's better to know that it is in there. Plus, such structural details, as you will see, such structural details can also help to do a type of fingerprinting of my lignins, right? If we want to do this green transition, and if we come to the point that the lignins become the starting materials for all sorts of things, hopefully, maybe one day, then eventually I want to know where my lignin comes from. What type of process did it see? Maybe I do not have that information anymore. If there are lots of suppliers providing lignins with no name, with not a clear history, maybe a mix of lignins, etc. So being able to develop a fingerprinting type of analysis system for identifying the type of lignin, its history, gives me a better chance to not choose the wrong lignin for my intended application. Not sure whether the point is clear, right? But I wanna get to a lignin analytics that allows me to do to also understand, to get information out that some people might try to hide in order to sell their stuff for a higher price, etc. Because, as I said, if the lignin enters the market with all the rules that we have in the economy, we will sooner or later end up in this position. So better to get prepared. So I propose to do always a type of fractionation to see then in detail what happens. So the first thing where we applied this was the craft lignin together with Claudia Cristini years ago. And then by the analysis of the various fractions, we got a picture of all the interunit bonding motifs, obviously, right? And we found some interesting aspects in terms of end groups, right? The lignin chain, the lignin polymer, the lignin oligomer does not always end up, obviously, in a phenol, right? We can end up in a saturated, in a cinnamyl derivative, or in a where we lost the double bond, for example, or we end up in an acid, or we end up with a typical cinnamyl type of motif, etc. All this makes a difference if I think of a valorization, because this is an active aspect, a reactive part. This is a non-reactive part, so I need to know. So by doing the fractionation, playing with solubilities, etc., I can enrich different types of motifs in different fractions, and I make them visible in my NMR analysis, basically. And then at the end of the day, I can come up also via an analysis of extractives, identifying molecules that are not lignin, but that are in the lignin due to the process, etc. I can come up with a cleaned up structure and a very, very useful picture of the structure of my lignin for valorizations. And then in craft lignin, the interesting thing was at the end of the day that probably it is not very homogeneous in the terms of the size and in the molecular type that we have in there. We might have at least two fractions, one fraction of smaller molecules, more condensed, one fraction of larger molecules, less condensed. And normally those from a reactivity point of view, you can imagine that this is chemically not the same as this, right? We have the same atoms, we have the same functional groups, but in a completely different correlation. So this is something else than this. In a lignin, without fractionation, it comes together, I get an overall picture, but the overall picture doesn't help me if I want to devise the perfect valorization, actually. Now, we applied the same technique then later to a withdraw lignin. Also here we can do fractionations. We did different types of fractionations, and in one approach, we managed to get the idea that also wheat straw is rather oligomeric, not polymeric, and different in structure. It's small, not as large as was thought before eventually, and it's not condensed, right? This is completely different from a chemical point of view than this. I mean, this is obvious, right? Just by looking at the structures without even looking at the details. So obviously a wheat straw and an organosolv lignin, right? This is the same picture as you will see we get also for wood lignins. It's completely different than a craft lignin, right? And this is very, I mean, should be like this, but that it is so different, right? That it and then seeing how it is different is also thanks to the NMR analysis that we can do. Now, then if we change a little bit the fractionation technique and we run our standard set of analysis, then you see that then you can even isolate nicely and prove that there are the LCCs, the lignin carbohydrate complex fractions. And seeing that so clearly in one of the fractions and not in the others would give us the possibility, if you think it in a different direction, to purify the wheat straw from the LCC. So if we do not like the LCCs to be in the wheat straw, or even better, if we want to use the LCC fraction for a valorization maybe in the biomedical field, because we like the fact that we have sugars attached to the phenol, so we have, you know, the best of both worlds practically in one molecule attached to each other, we can isolate it. We know, thanks also to our analysis, that we can isolate this, we valorize it in way R, in way A, and we use the different types of fractions that we have, and we use them for, I don't know what type of application, okay, where it is best. So so the analysis of the NMR gives us a hint to understand, as I said, impurities. This would be, if without fractionation, these little peaks here would not show up in an HSQC of the bulk material. It's very easy to not see them, okay? So fractionation, as I said before, fractionation gives us a possibility to see structures that are hidden in the bulk, but that are eventually essentially for the performance of the material if we want to valorize it. Now, let's do it a third time. So we take a birch lignin. Now we have different, this was done together with the group of Christakopoulos and Matsakas at LTU, for example. We isolate, they isolated the lignin, and then we did the analysis. And here we didn't do a fractionation, but we went directly for the bulk material, but we compared now on a very deep level the bulk material from different extraction processes. And at the first glance, you see that all looks the same, right? This is the quantitative, we use the technique in this case where we have the quantitative carbon and the normal HSQC. So we quantified according to the quantitative carbon and correlated it to the signals. You see, there's not much difference, but the longer you look at them, even on this size, you see that there are subtle differences between the various extracted lignins. And at the end of the day, the analysis combined with the phosphorus that I do not show you here gives us the possibility to understand which single effect has a single process parameter on the lignin structure. For example, if we increase time, we get smaller structures, and that is a very general thing. We get cleaner structures, less clean structures according to process severity, etc. Especially this last point, that we can eventually use the phosphorus and the HSQC NMR in order to also identify process severity indicators, work very well if we looked at the structure at the analysis of an organosolv pre-treatment of a softwood of spruce. Here, for the first time, we managed to identify something that we call then at the end of the day human lignin hybrids or lignin human hybrids. Because here the LCC, the analysis revealed basically that there are some cross peaks that are surely not sugars, that are surely not lignins, and they are not extractable. If you remember what I showed you for the, I showed you in the here, no? I said for the softwood kraft lignin, we did an analysis and trying to find out what is extractive. So what is in the lignin that can be easily extracted by just a simple wash with ethyl acetate, for example, right? Here we can do that, and we can just disregard those peaks for the lignin analysis. If we do the same here, those peaks do not go away. And we have very distinct cross peaks. And then, thanks to all the literature that is out there in terms of lignin, furfural, and lignin and furfural phenol resins, for example, we managed to identify that also in this lignin, in this softwood organosolv lignin, based on the process or as a function of the process severity, we might have lignin human hybrids. Okay, so we do all the analysis, classical type of analysis. We identify the cross peaks, we identify the functional groups, more or less what you would expect, nothing too surprising, etc. But if you do all this, you remain with clear peaks, and these clear peaks is not rubbish. It cannot be washed out, so it must be connected to the lignin itself. It's not a fraction that can be fractionated. It is there, it is connected to it. So that means it must be covalently bound. And then if it's covalently bound and it classifies and it corresponds to cross peaks of humans as they are reported in the literature, we must postulate at this point that in some cases, something like a human lignin linkage exists and that we have hybrid structures which are not lignin, in the lignin. So we basically, according to as a function of the process parameters, the NMRs, the analysis revealed that we can have something that is a natural polymer in the sense that the ingredients come from nature. Now, one is a sugar-derived one is the lignin-derived one, but then we've joined them together in our processes. So we have a completely different thing. It's not lignin, it's not human, but it's a copolymer, basically. Okay? To a certain extent, we have a copolymer in there. And also that obviously affects our type of valorization that we can run eventually. Now, the interesting question is this a general thing, right? So what we did is then going on and looking at other organosolv lignins, and then maybe for all of you that are doing organosolv, I can assure you it's not a general thing. It is really process parameter depending. And what you can see is, for example, that in wheat straw, we don't have the signals. We have eventually, there are some regions here where we have to check, basically. Okay, so the HSQC says us, for example, that that could be a cross peak, that could be one of these indicators of these hybrids. Then taking the NMR for the carbon, we see that the corresponding other peaks that fit to the structure, like this one, like the quaternary ones here, for example, that they are missing. So we don't have them. So the structures are not in there. So it's a nice combination. Obviously, the phosphorus in this case, even if I don't show you the phosphorus spectra for these structures, without the phosphorus data, guys, we don't arrive to these structures because we need to correlate whatever we see as structural unit in the HSQC, obviously, to our phosphorus data. Now, the numbers of OH groups need to fit. We cannot have a huge amount of phenolics and they say we have a completely polymeric structure, right? The more phenolics we have, the less it is polymerized. Otherwise, the phenolics are occupied in the chains, right? If we compare it to the ratio to the abundance of the aliphatics. Now, so why are we doing all this? As I said already, I personally like to do this in order to have a better idea of what valorization works, right? So then, in order to answer the questions from the very beginning, so what can I know? I can know all analyzable aspects, not only the ones that I talked about from the HSQC and the phosphorus, but I should take into account all the others, no GPC, thermal analysis, etc. Because with the humans, a nice cross proof was a thermal analysis, a thermal stability analysis. Then we can have all the chosen analytical techniques that can tell me this is what I can know. My analytical tool kit basically allows me to see something. Then what can I know? I know as much as I am willing to understand and what I'm looking for, right? If I close my mind and say, this is lignin, I do not even look for something else, I end up with lignin, but it's completely wrong, probably. And then what can I really know? Well, guys, you know, as much as we advance with the analytical techniques, our picture of the lignin already changed drastically. It might still change, right? We are far away, as I showed. When you look at the structures that I showed you and you look at this old structure here in the background, we made a nice step forward, right? So we can do much more. One thing I like to do is to put all these analytical data together because I want to arrive at a lignin database so that I have 3 key data, and then the computer tells me you probably have this lignin and this is the best way to valorize it, right? That is my personal driving force for doing this NMR analysis. So with this, I would like to thank and close. I obviously thank my group members here. I thank the principal group members at LTU. And I obviously thank all the postdoc, PhDs, and master students that suffer working with me, and the guys that allow me to pay for the chemicals. Thank you very much.
Thank you so much. Thank you, Heiko. Can you hear me well now? Yes? Very good. We have a question directly from Fabio. And I was thinking that maybe I can allow Fabio to speak. So let me see, allow to talk. Yeah, Fabio, can you please tell us your question?
Do you hear me?
Yes.
Yeah. So, Heiko, first of all, thanks very much for the nice presentation. I think this is the first NMR presentation I can follow from the beginning to the end, and I don't work directly with it, so thanks a lot. I have 2 questions, if I may. First one is, I think I missed the connection between the phosphorus NMR and the HSQC, because at certain point in your talk, they were sort of overlapping somehow. So if you can quickly comment on that.
Yes. So, I mean, the phosphorus NMR is, so the phosphorus NMR is for analyzing the OH groups in the structure, right? Let me call it, I recall a slide and then I explain. So, for example, if we take, do you see the slides?
Yes.
Okay. So if we, sorry guys. So if you take a lignin structure, right? We have, in a lignin structure, we have the OH functionalities, which are very, very important because the phenolics give us all the characteristics of the polymer as such, right? And then we have the aliphatic OH groups, which might give us a handle for functionalizations, etc. So these OH groups can be seen in a proton NMR, but they are not clear in the proton NMR. For that, we run better a phosphorus NMR, so we can nicely get a picture of a qualitative and quantitative picture of the OH groups in the lignin. Okay, only the OH groups.
Okay.
Then for seeing all the rest in terms of, you know, the aromatic ring structure, the aromatic rings, the aliphatic units that are connecting basically the single aromatic rings, we run an HSQC.
Okay.
Because in the HSQC, we see only the carbons that are connected to at least one hydrogen. Okay. So in the HSQC, we see, for example, this carbon here, because it is an aromatic ring carbon that carries one proton, right? One hydrogen. We will not see, for example, this carbon here, because this carbon here is only connected to other carbons and to the methoxy group and not to a hydrogen. So in the HSQC, we get practically a picture of all these linkages, right? And we can, and that allows us to identify, for example, this type of structural motif here, or this type of structural motif. So we get a, wait, I go to another slide. So we get a picture of the type of linking motifs that we have in the lignin.
Okay.
Okay.
Okay. Yeah.
We do not see all the atoms. I mean, in the phosphorus, we see the phosphorylated OH groups. Okay,
yeah.
And we exclude all the other stuff. In the HSQC, we exclude the OH groups, and we exclude all the carbons that are not connected to at least one hydrogen. Okay. So in both cases, we simplify the vast amount of structural motifs that we have to a single feature, let's say, right? In the phosphorus, the feature is OH, that becomes OP, O phosphorus. And in the HSQC, we go for groups that carry at least one proton, one hydrogen. Right? Then if you want to see all the carbons without having interferences with the hydrogens and the oxygens, we go for the quantitative carbon that I mentioned, but I didn't put too much emphasis on it, right? So we try, and you get the point, right? We try to reduce complexity by choosing the right analytical method for targeting one of the features.
Yeah.
Then we need to recombine it essentially, right?
Can I ask another question, or there are other people?
You can go ahead, and then we will take the next questions.
Yeah. So the other question is more a practical one, because to increase resolution of the signals that we see, especially if we have overlaps in the HSQC, you said that it's very important the sample preparation, like also fractionations and analyzing the different fractions you get. But is there also another option if you cannot work with the sample preparation? Because, for instance, we use this analysis to detect the LCCs in spruce after enzymatic hydrolysis, and we get a messy region in the HSQC because of the old overlap, but we don't have enough sample at the same time, so we cannot work on the sample preparation. I was wondering if, let's say, the sequential HSQC, if we can call it like that, can be helpful, or you have other tips?
Well, I mean, the HSQC 0, this back extrapolation, you know, you can do it when you see the signal. If you're doubtful whether there is a signal or not, then you know, if you even in a standard HSQC, you hardly see the signal, then the extrapolation will not help you, because you will lose the signal in the other two.
Okay.
So in that case, the only possibility is to run many more scans, right? So you have to eventually go, I do not know what NMR machine you have. It's a 400, 600.
No, I have no idea, because I'm not the one actually doing the measurements.
Okay, so the only way to get a better, I mean, you need to work on the signal to noise ratio in that case, and the only way to do that is to increase number of scans.
Okay, thanks.
I mean,
this is a tips I can suggest.
Yeah, well, I mean, unfortunately, if you cannot fractionate, and if you cannot concentrate the sample, it's the only way. The other way, obviously, right, running more scans on the same concentration of sample that you used so far, or you try to concentrate the sample.
Okay, yeah, makes sense.
Right, and concentrating the sample is not trivial, however, because you easily run into solubility issues. So one trick that you could use is, there are two tricks. One trick is to use to do an acetylation of the OH groups, because that increases solubility. Okay.
Okay.
The other, and there are protocols for that. The other trick is to play with the solvent system that you use for dissolving the sample for the NMR. So if you by default use it, for example, if you use DMSO.
Yeah, DMSO.
Yeah, then you can eventually try to mix in other solvents to work with binary solvent systems. If you really have problems on solubility, you might even play with deep eutectic solvents or ionic liquids as a solvent system, with the effect, obviously, that you bring in the peaks from the solvents, right? So then it gets a little bit messy. But if you look specifically for dedicated peaks and you manage to find a solvent system that in the area where your interesting peaks are does not show up, you can do that, right?
Thank you very much. Very helpful. Thanks a
lot. No worries. Feel free. I mean, if you, I don't know, I mean, somehow it should be possible to contact me or Treesearch, and then we can get in contact and we can talk later.
Thanks.
Also.
Thanks a lot. Appreciate it.
No worries.
Yes. Thank you so much, Fabio, for the nice questions. Then we have two more people with the questions. The first one was Mikkel. So Mikkel, would you like to unmute yourself and ask the question?
Yes, thank you. Thank you, Heiko, for a great presentation. I really enjoy it. I'm also in my spectroscopy, although not in the lignin fields, trying to get there. So the general questions I have is like, how do you obtain connectivity between your different units? Because within the unit, yeah, I could kind of get, you may employ HMBC or other type of experiment, but within the unit, maybe it gets more complicated, because your HSQC only gives you a pair of different types of carbon-proton pairs, basically. So how do you get that connectivity and get that bigger sort of picture of your lignin?
That's a very good question. So what we actually see, and that's a really good question, because what we actually see, if you see the slide now that I have here, what you actually see in the HSQC, you can say what you really say on the basis of the HSQC is that you have this structural motif, for example, right?
Exactly, right.
Because you see essentially, I mean, the dotted carbons in the chain here are the signals that are most relevant, or that are used also for quantification. I also see the third one, this gamma position here, right?
Right.
I see that, but I normally tend to not use it for quantification, because the gammas are extremely difficult in the HSQC. Okay, even if you run it quantitatively, they tend to make trouble. Okay. So we see, what we know is that we have this connectivity for this inter-unit linking. We have this type of connectivity, etc. What the HSQC does not tell us is whether, for example, this one is followed by this one and then followed by this one.
Exactly, right.
That is something I cannot, from the HSQC, I cannot deduct. Okay? If there's no NMR technique that will give you that information. This is a type of information that we need to get out from a high resolution mass spectrometry analysis. But even in the high resolution mass spectrometry analysis, it will be, I mean, people try it, okay? And there are huge discussions about that. Whether then the fragments that you see in the mass after ionization, whether you can know that come up with a certain mass, whether that is really indicating a sequence like ABC or a sequence AA, BA, etc., etc. Okay, that is really tricky. That is something that people are currently working on. So the structures that I show you now, when I show you then a structure, that is a statistical representation of the functional groups identified and quantified via the HSQC. Right? If I see, for example, 20% of this beta 04, right? then I try to come up with a structural proposal where I have 20% this group, 10% of the other group, etc. So and then I normally draw more than one structure in order to have at least an idea of how can be distributed the different percentages that I take out from the quantitative analysis in form of a structure. We made attempts where we were working on a computer-based description, very, very flexible, of lignins. And if you feed the computer with the percentages of the different inter-unit linkages, it comes up with 10,000 of structures that meet the statistical requirements, right?
I was thinking about that, yeah.
Yeah, no, it's really amazing, right? When you look, then you sit 1 hour in front of the computer, you click, 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 and you see thousands of structures that are possible structures that fit your analytical data, right?
Yeah.
That gives you a completely different view of your material, obviously, right? And that is something very nice, to be honest, right? But that is something we work on, and maybe sooner or later I will manage to finish that. But it's really we will manage. We should say we, because I do it in collaboration. We should manage to finish that. It's really intriguing, honestly, right? But this is the man-made, the simple version of it, in order to get at least a type of idea of the structure. But as I said, the sequence, but A, B, C, A, B, etc. is out of proof at the moment.
Great. If the time permits, may I ask a quick follow-up? Another question.
Is it okay for you, Heiko? Heiko, is it okay that we take...
Yeah, I'm fine.
Okay, okay, great. Yes, great. Please go ahead.
Yeah, so I got interested in this quick quantitative HSQC, the QQ HSQC. And I've been running some HSQC quantitative from the Helsinki group. I think they base on this QQ HSQC experiment too. Are you aware that Bruker has that in the sequence? And
yeah, no, Bruker doesn't have it.
Okay.
You have to, the sequence is published though.
Okay, so I can obtain that from the authors.
Yeah.
Hmm.
To be honest, I mean, it's not the author, the professor, the group leader has the publication, and then he has an online repository where he has the sequences.
Okay.
So you take the basics of the sequence from his website, and then you adopt it to your specifics, to the specifics of your instrument, obviously, right?
Yeah,
so
exactly.
You need to be able to program the NMR, otherwise.
Yeah. But he doesn't provide like a Bruker pulse sequence right from because then I could just quickly adopt it. Maybe even he has parameters.
Well, it is based on it is based on a Bruker sequence, right? You just add add the additional pulses, etc., in order to get rid of the back coupling artifact, etc., etc. So it's you start. But I mean, he published the sequence. You download the text file which contains the code.
Oh yeah,
and
exactly.
then you adjust the powers of you adjust that you normally adjust to your instrument, like power level, etc., and that's it.
Yeah, no problem. I mean, once I have the code and even parameter files, I can then translate it into my.
As I said, I mean, he has an online repository where you can access it. It's not easy to find, but if you contact him, he will get back to you.
Okay, thank you. That's all. Thank you very much, and thank you for patience.
No worries. You're welcome.
Great. Thank you for the nice question, Mikkel. And Heiko, do you still have time to answer one more question?
I'm fine. Don't worry. I'm fine.
Rustam, are you still here?
Yes, I'm here. Thank you very much.
Please go ahead.
Thank you very much, Heiko. Amazing lecture. I have a question about fractionation. You mentioned your dialysis packs. So generally comparing other methods for fractionation of lignin, for example, using different solvent or acidic, pH-based method. So I think dialysis pack can be quite pricey. I might be wrong here, but generally, what method would be your go-to for lignin fractionation?
Well, the easiest, I mean, the thing to start is probably choosing a standard set of solvents that have different, in order to play with the Hansen solubility parameters, basically. Okay, so you take, for example, 100 grams of lignin and you throw it into acetone. You stir it for a certain amount of time, and then you filter off whatever is not soluble, and you start from there. Okay, yes. So for example, what I mean here, sorry, if I find it, just give me a second. Here, for example. Okay, So we did different. I mean, here we played around with different mixtures of acetone and hexane, for example, in order to stepwise reduce the polarity of the solvent system, right?
Yes.
So for the next approach here, we played around with the H bonding character of the solvents that we use. So a donor instead of an acceptor, for example, right? The acetone would be an acceptor. Then if we use methanol, we go for a donor. So that gives you different types of interactions with the lignin molecule, and you separate. It is not, I mean, maybe you cannot predict at the very beginning which type of fractionation is best, right? But you need to, in order to start from somewhere, I would always start from a simple solubility program, let's say. So take the lignin, take one solvent, and then do a first filtration, and then treat the solid fraction with different solvents, or try to precipitate something from the soluble fraction.
Okay.
In order to get a first impression, let's say.
Because
Acetone, hexane.
Because I suppose, for example, with acidic fractionation, it can also interfere and change the structure. So probably not the best way to do the fractionation. So solvents and then dialysis bags, I guess.
Well, you have to. I mean, yeah, dialysis bags. I mean, we did it. I mean, for the fractionation, we worked mainly without dialysis bags, because, as you said, it's costly, right?
Okay.
So what is easier is really this the classical setup. Beaker, you put in the lignin, you play with the solvents.
Okay.
That is, well, if you want, you could also go for a reactivity-based fractionation. If you use a solvent that is not innocent in terms of chemical reactivity, that might give you completely different insights, right?
Yes.
You could do affinity chromatography in order to take out the acid-containing groups with a base resin, for example, right? So that is depending on what you want to see, you devise your fractionation technique.
Okay, thank you very much.
I would always go for a blunt fractionation and then take a fraction and play with the fraction. That gives you the best.
Okay, yes, thank you very much. And I might have missed it in the beginning. Is the recording going to be available somehow? Oh, okay.
Yes,
Thanks.
we are planning to upload it on the YouTube. Hopefully everything goes well now. Yes, and it will be available on our YouTube channel. Rustam, can I ask you, which university are you connecting from, or organization?
I'm currently at TU Vienna in Austria. In Vienna.
Good, because I didn't find you in our Treesearch participants, but really nice. Thank you for joining. Yes.
Thank you very much. I was very happy to hear about the free webinars, and it's very useful. Thanks.
Yeah, you're welcome. Just go to our YouTube channel of Treesearch, and there you can see the recordings. This one will be available also shortly, in a few days.
Okay, thank you very much.
Yes. Good. So are there any more questions? Last chance. I don't see any other questions in the chat, and we already took some extra time. So I really would like to thank you, dear Heiko, for the great lecture. It was also really useful for me. Yes, thank you so much. Gunnar is also sending you big regards. He had to run to start teaching.
No worries, I know the problem.
Yes.
I have exams now. I will do exams now. So.
Okay. Good luck to you. And I would like to thank all participants. Thank you so much for all the great questions. And please stay connected. We have this year 2 more webinars. So on the 29th of November, it will be DNP NMR by Lars Evenes. And then on the December 13th, Thomas Larsen will be talking about the CP mass NMR of the isolated cellulose. So thank you so much for today, everyone.
Thank you very much. Bye-bye.
Bye-bye.
Bye.